Hello, and welcome to today's webinar, One Woman's Journey in Cancer Research. I am Dr. Barbara Malkas, Board President for Massachusetts Breast Cancer Coalition, and I'd like to thank you all for joining us today. Massachusetts Breast Cancer Coalition is dedicated to preventing environmental causes of breast cancer through community education, research advocacy, and changes to public policy. I am very pleased to welcome today's speaker, Dr. Linda Malkas. Yes, and my sister. Dr. Linda Malkas is the Associate Chair and Professor in the Department of Molecular and Cell Biology at the Beckman Research Institute of City of Hope. Her major research focus is the relationship between DNA replication and cancer. Her laboratory's recent work has led to the opportunity to develop a truly selective therapeutic agent that will inhibit proteins uniquely expressed by cancer cells. Dr. Malkas, welcome.
Hi, very nice to be here, and thank you for the opportunity to participate in this. So as my lovely sister, with the lovely introduction she gave, we hail from New York. It'll probably show up in my accent. So we grew up in Queens, New York. So today I'm going to talk to you about a journey that I've been on that ultimately led to the discovery and development of a brand new anti-cancer agent. I know that you are focused on prevention and environmental causes potentially for breast cancer. But this agent down the line, we think that it could also play a role in prevention. But I'm going to talk to you about how we got here. Starting my talk, so I'm going to call it Linda's science story. And I always tell people that when you see me, you're actually looking at Alice. And I just followed the science rabbit down the hole. Originally, I never thought I would become a cancer researcher. That was not on my scope. In fact, I wanted to be an astronaut. That was my first thought. I was going to be a space scientist. At the age of 9, I dedicated myself to that. And I actually started in engineering school. But then I took this course called organic chemistry, which 99.9% of the people who take that course hate it. But I loved it. It's the study of carbon, and we're carbon. And the beautiful thing about organic chemistry, it allowed me to understand how the very, very complex molecules like DNA, how they actually form. It's almost a miracle if you think about it, how these nucleotides come together to form these very long strings of DNA. And that's entirely driven by chemistry. That beautiful shape, I'm sure most of you have ever seen the structure of DNA. It's what they call a double helix. It's very, very beautiful spiral type of structure to it. And that's totally a chemical driven process. And just to show what kind of miracle we are, and get the idea that, each of us are way more than we think we are. If you took one human cell, which you can't even see, and if you look inside the human cell, there's something called the nucleus. Don't worry about it. But inside the nucleus of each human cell, I call it the house of DNA. That's where DNA lives, inside the nucleus. And if I was to take the DNA out of one nucleus, out of one human cell, and I was to stretch it out, it's actually 3 feet long. And one of the great mysteries is how there's a lot of mysteries. One is, how do you roll up 3 feet of DNA and tie it inside of a tiny, tiny little nucleus? How do you do that? And people are still studying and figuring out how that's done. But also, every time a human cell has to divide, and you have ongoing cell division all the time. Your entire gut turns over in like 2 to 3 days. So you have an enormous amount of cell division going on. And every time a cell has to divide, it has to synthesize a whole new complement, 3 feet of DNA, to give to its daughter cells. And that's a process people are still studying. There isn't a human machine that we have made that can come anywhere near what the human cell can do in the act of DNA replication. And a cell will replicate 3 feet of DNA in about 8 hours. This is a massively fast process that occurs with great, great fidelity. So I just wanted to lay that all out for you because that's been my world for the last 25 years. So here's Linda's story as she followed the science rabbit down the hole. So what I'm going to do is talk to you. I don't want this to get too scary, okay? But this does represent about 22 years of my life, this talk here. As I said, I never planned on being a cancer researcher, but here I am living in California. So I'm going to talk about the discovery of a new molecular target, how we characterized that molecular target, how we discovered and developed an extremely novel new anti-cancer drug, and how we are moving it forward. So when I was talking about the act of DNA replication, I hope you can see my little cursor moving around. And so I'm over on the left side here, and there's something called the human cell DNA replisome. So in order to carry out the replication of 3 feet of DNA, it actually is done by using a machine. The proteins, these little cogs that are involved in the replication of 3 feet of DNA, come together to form a molecular machine, which we call the replisome. And they got the name of some of the cogs inside of this machine, this very beautiful, elegant machine. And long time ago, when I was a brand new assistant professor at the University of Maryland School of Medicine, I was reading a paper, and it's a very cool paper. It was written in the 1970s by a fellow by the name of Lawrence Loeb, Larry Loeb. And it was an interesting paper, a science paper, and then it didn't really have data, but he proposed something in it that tickled my brain that led to where we are now. And what he said was, you know, cancer is, and actually all disease, if you think about it, all disease is because of a change in DNA, either a mutation or some kind of damage to that beautiful molecule. But disease is because of a genomic change. So I had way back when I was in Worcester, Massachusetts at the Worcester Foundation, I was a postdoc there. I had found and discovered and published on the first isolation of a molecular machine, which is this little machine over here on the left side. I was able to isolate from human cells for the first time the DNA replication machinery. In that I could isolate it from a human cell, stick it into a test tube, offer it DNA, and it would create new DNA or synthesize new DNA just like it would do inside the cell nucleus. And that led to my getting my first faculty position in the University of Maryland School of Medicine. And I'm reading Larry Loeb's paper, and I'm thinking, gee, if cancer is the result of a change in DNA, and I'm the mother of a DNA replication complex, a DNA replication machine, I wonder if that machine has changed in cancer. And you know, it's funny how the simplest of questions can lead to some really profound answers. And down here on the left side, we created an assay or a method for evaluating the act of DNA replication. And we call it a DNA replication fidelity assay. And what we did was we isolated the molecular machine, the DNA replication machine, from either malignant or non-malignant breast cells. The non-malignant breast cells down here are the yellow and green. This is the replication machine from non-malignant breast cells. And then we also isolated the machine from malignant breast cells, which are these guys here with the higher bars. We measured their ability to be faithful in replication. And the first thing that popped out is that the machine that you isolate from non-malignant cells, the yellow and green here, that machine is very faithful. It doesn't make mutations, not readily. Whereas the machine that you isolate from breast cancer cells with the higher bars do not carry out replication faithfully. They make mistakes. So that was interesting. That led to a lot of press, so to speak. I knew who a lot of the cogs were that were inside this replication machine. And we started looking through all of these different proteins for who was different. And we found one that was different between non-malignant and malignant cells. And it was called proliferating cell nuclear antigen, or PCNA. And this is how we found it. We performed something called 2D gels. Don't even worry about what it is. All it's doing is looking at a change in charge on the protein. In non-malignant breast cells, the form of PCNA, okay, has a very basic, this is the acidic side of the gel, this is the basic side of the gel. The non-malignant form of PCNA has a very basic charge on it. When you looked at PCNA now in malignant breast cells, they too have a basic form of the protein, but now there was new acidic form as well. We then, I struck up a collaboration with a wonderful investigator named Lauren Schnapper. She's a breast cancer surgeon down in Maryland. And she gave us a whole lot of tissues. And she blinded us in the study. So either it was non-malignant breast tissue, benign breast tissue, or malignant breast tissue. And we did all these gels to measure the charge on it. And when we cracked the code, or she cracked the code, what we found was everywhere there was a diagnosis of malignancy, that tissue had this funny form of PCNA, or the acidic form of PCNA. So we had found something that was different in a cancer cell versus a normal cell. Cancer cells contain an acidic form of the protein PCNA. Now, a little bit about PCNA and why it's so important and why it led to such a very interesting drug. PCNA, you can kind of think of PCNA. Now, this is the structure of PCNA. I hope my cursor is going around here. So what PCNA does is it forms a ring. And what it does, it encircles DNA. And I always liken it to like a shower curtain ring. You know how your shower, so if you can think of the shower curtain rod, all right, in your bathroom, your shower curtain rod is like DNA. PCNA is like the shower curtain ring that circles around that rod. And so DNA would be here in this circle, and around it, this protein folds itself around DNA. And what it does is just like a shower curtain rod, or a shower curtain ring, it can slide along the rod. So it slides, it's called a sliding clamp protein. Clamps around DNA and it slides along the DNA. And that's interesting. So that's what it's doing. It's sliding along DNA. But out here, where the red box is, it does something that's pretty amazing. It interacts with about 200 other proteins inside the human cell. And all of these other proteins have very critical functions. They of course, are involved in DNA replication. They're also involved in DNA repair, cell cycle control, chromatin assembly, all kinds of things. So this protein, this little sliding clamp protein, actually has to orchestrate how all of these other proteins interact with pCNA and work on DNA. So that's a little bit about pCNA. The other thing about pCNA is that it is involved, pCNA is involved with about every DNA repair process that we have inside the human cell. It is orchestrating the repair of DNA, not just the replication of DNA, but the repair largely is involved in just about every process that fixes DNA. So here we had found this funny form of pCNA that was in normal cells versus malignant cells, right? It was all in breast cancer cells at that time. So what we wanted to do, okay, so you can say, well, okay, there's that 3D structure over here of pCNA. Now, we didn't know what made the basic form from the acidic form. What was that? And I won't bore you with it, but it took about 4 years to figure this part out. We knew what the amino acid structure, what all the amino acids that form this protein are, okay? But we didn't know where on that protein, what was different between the acidic and basic forms. And I won't bore you with 4 years of work, but the bottom line is that we discovered that the change between the acidic and basic form of pCNA occurred between amino acids 126 to 133. Don't stress too much about it. Just know that we identified the address of the difference between this acidic and basic form of pCNA. And what we did was, once we had that address between 126 and 133, we created an antibody, okay, that only recognized that funny form of pCNA in cancer cells, and that's shown here. We created an antibody when it was between 126 and 133 that only recognized that acidic form of pCNA and not the basic form of pCNA. And so we thought, wow, this could be a great tool. I wonder if it could be used by pathologists. So this is an antibody, and we gave it to a pathologist. And what you're looking for is brown cells. So this was a test with this antibody, and to see if this what we were calling now the cancer-associated form of pCNA, that acidic form of pCNA, was it really a phenomenon of cancer cells? So using that antibody, we gave it to a pathologist by this, his name was Sunil Bobby, an absolutely outstanding breast cancer pathologist. And we said, okay, Sunil, we think this is really something that could be helpful to you for identifying breast cancer cells. And he goes, oh, yeah, he didn't quite believe, like, yeah, sure, he's seen everything. So he went away, came back 3 weeks later as like my new best friend. And this is what he showed. So what you're looking for are brown cells. If our antibody against that acidic form of pCNA or the cancer-associated form of pCNA was in fact recognizing cancer cells, it would stain the cancer cells brown. All right, so that's what you're looking for. So here you're looking at normal breast tissues. You can see all the little ducts and everything in here. And you can see that there are no brown cells, right, when it's stained with the antibody. If you look at DCIS, you can see some light brown cells. Okay, that's ductal carcinoma in situ. If you look at invasive breast cancer, oh man, you can see a lot of brown cells when stained with our antibody. And it could even pick up a metastatic lymph node. So we can see here, this is a lymph node from breast cancer. So we showed with this experiment that 126 to 133 region of PCNA, the PCNA isolated from cancer cells, that it was very specific to cancer cells and not to normal cells. Well, we had done all our work in breast cancer at that point, and it became a cottage industry when we were at the University of Maryland School of Medicine to start looking, is it just a breast cancer problem or is this a cancer problem, this change in PCNA? And using our antibody against now, which we were calling the cancer-associated form of PCNA, we started looking at all kinds of cancers. And everywhere there was a diagnosis of malignancy, they harbored this this acidic form of PCNA, this cancer-associated form of PCNA. So I was moving along in my career. I was getting a lot of visibility. We had identified, that the replication machinery of cancer cells was altered. It was more mutagenic, more error-prone, and that we had identified a component of that machinery that was selectively changed in cancer cells versus normal cells. And I thought my career would just be going along. At this point, I became the Vera Bradley Chair for Oncology at the Indiana University School of Medicine. And I created a breast cancer program that stretched over the entire state of Indiana. I was very proud of our work, and I really thought I was going to probably live in the Midwest forever. I fell in love with the people of the Midwest. And I'm going to give you a little personal story because this actually was, and I have often said, I hope that everyone has a moment in their life where everything falls into place and you know what you're supposed to do. So, sidebar, personal story. So anyway, as I said, I built this breast cancer program in the state of Indiana. I was moving along in that. In Indiana, the big newspaper there wanted to do a story on me, and they were sending a photographer to come take my picture. And so we agreed on the date and time for the photographer to come take my picture. And well, anyway, the date and time comes, and there's no photographer. And half hour goes by, no photographer, an hour goes by, 2 hours go by. Okay, and I'm a New Yorker. Okay, patience is not one of our virtues. And needless to say, I was pretty royally pissed, but he was not there. And so I go back, I go into my office, I'm working. Eventually, this man shows up in the doorway of my office, and he looks like a wreck. He's like in his late 30s, but he is dropping his cameras. He looks like a wreck. And he is, Dr. Malkas, I'm so sorry. I'm so sorry. And I'm so glad I did not act like a New Yorker. I am so glad. But anyway, I looked at him and I went, "Are you okay?" And he said, he was going to say yes, but then he said no. He said, "My daughter is across the street at the Riley Children's Hospital. She's had neuroblastoma since she's 3 years old. It's been 5 years and we're running out of options." And well, that just took the steam out of me. Well, anyway, we go take a picture, and it's a beautiful picture. I still use it on LinkedIn. And even though it was decades ago, but apparently you can use old pictures on LinkedIn. But anyways, he's putting all his cameras together. He says, "Dr. Malkas, what is it you do?" And I don't know what it was, but I looked in this man's eyes, and I realized, what do I give him? What can I give him? And I realized I didn't have what can I give him except, one or two bright ideas I have in the work of these hands. So I said, "Come here." And I had him sit by me in my office by my big laptop computer. And for 2 hours, I gave him the only thing I could give him, which was my data. I told him about 3 feet of DNA and this replication machine and how it was changed in cancer. And I gave him charts and graphs and tables of numbers. And after 2 hours, he said, wow, I really wish you good luck, Dr. Malkas. And his name is Steve. And I said, I wish you good luck too, Steve. And he went away, but I kept tabs on his little girl. And she went, not long after I had met him, the little one died. She had just turned 9 years old. Not long after that, he calls me in my lab and he says, my wife hasn't left the house. Could I come to your lab? And I thought, okay. And I prepared my lab to have a beautiful lunch. And he came, and all my young people in my lab talked about their research. And then he did something that changed the course of my life and why I'm even talking to you today. He said, the last time I was driving my daughter Anna to the hospital, I was looking at her in the rearview mirror. And can you imagine? And he said, Anna, what should we do? And this little 9-year-old says to her dad, more research, Dad. At which point he handed me a check for $25,000. Now I had grant money, but this was secret money. And I wanted to do something bold in the name of this little girl. So I got this idea, which was crazy, okay? And I said, gee, I got this protein, PCNA, that's only in cancer cells and not in normal cells. I wonder if I could make a drug. It's crazy because I don't even know how to do this. I wonder if I could make a drug against this funny form of PCNA in cancer cells, because if it works, it would kill the cancer cells, leave the normal cells alone, and it would be way less toxic or not toxic at all to a patient. And wouldn't that be great? So in Indianapolis, there's a very large pharmaceutical company there. I won't name it right now. But I went over to them and I danced for these people. I told them all about this wonderful molecular target. And if they could make a drug against it, I'd work with them. And they were very kind and they listened to me. And after I got done, talking to them for an hour, they said this. They said, Dr. Malkas, that work you've done on the replication complex is beautiful, and identification of this funny form of PCNA is very elegant, but your target is undruggable. You'll never make a drug against this target. It has no enzymatic activity. No one will ever make a drug. Well, that was like hitting me with an arrow through my heart. And I left there and I thought, how could I have been so stupid? I went and I talked to these brilliant minds and all this stuff. But by the time I made the parking lot of that place, I was like, screw these people. I don't know how I'm going to do it, but I'm going to do it. And then not long after that, I got a phone call from a place called City of Hope in Los Angeles. And they wanted me to come out to be an external advisor. And when I went there, what they had done was they had put together a very unique infrastructure for an academic institution that would allow somebody like moi, who made an observation on a lab bench, they had put together all these people and infrastructure and facilities to allow somebody who makes an observation on a lab bench to move it all the way to the clinic. I was like, oh my gosh, I've got to work with these people. Then in the same time I'm out there visiting them and being their external advisor, he offered me a job and I didn't think twice. And I moved out to California in an attempt to make a new drug against an undruggable target. So as I have said, back to my story, right? We had identified now amino acids 126 to 133 as being an important address on this funny form of PCNA. We made an antibody which I think you can pick out now right here, right? You're looking actually at cervical cancer here, a piece of tissue, of cervical tissue, and your eyes can now pick out where the cancer cells are. So I said, okay, this is how I'm thinking. I'm going to tell you exactly how it is I came up with my thinking for how to make a new drug. So remember when I told you that PCNA interacts with about 200 other proteins, right? Now, I lived in the Midwest, okay, and in Indiana, but now I'm living in LA. But think about this. I learned that if you want to shut down most of the air traffic in the United States, what you do is you send a snowstorm either into O'Hare or the Detroit airports, because what you do is you shut down entire hubs. Now, in my mind, okay, I think of PCNA like an airline terminal. You see, I hope you see my little cursor. So PCNA is like this little airline terminal, right? And all those 200 proteins are like those proteins coming into those jetways. And I thought, what I need to do is build a snowstorm that blocks those planes or those proteins from landing on PCNA. And if I can build a snowball, a snowstorm that can take out this hub, it will block all those other proteins, all those other planes from landing on PCNA. And that was my approach. So this is what I said. What happens when you take out an airline hub, right? Remember, I told you PCNA is involved in almost all of the DNA repair processes and all other kind of processes in the human cell. I take out PCNA, I can shut down a lot of air traffic inside of the cancer cell. So it turns out now that if you look at what they call the crystal structure, the 3D structure of the PCNA protein, every protein has its own 3D shape. And if you look at the 3D shape of PCNA, it's got a lot of indents and every protein has it, okay? But if you look at the 126 to 133 region of PCNA, what you see is it makes a pocket. You're actually looking at the 126 to 133 region here of PCNA. And I thought, it's a pocket, huh? Suppose I can find a molecule or build a molecule or a snowball that would fill up that pocket. That pocket is involved in the binding of all of those 200 other proteins. And I could show that when I took the amino acid structures of a lot of the binding proteins and I lined them up for how they interacted with that 126 to 133 region of PCNA, what you saw was that all of those proteins, even though they themselves had different 3D structures, they all, in that one area where it was important for those proteins to bind to PCNA, they all aligned themselves to sit in that pocket. That gave me an idea of what a molecule had to look like to fill up that pocket. So I'm trying to figure out how to make a snowball to fill that pocket so all these other binding proteins can't fit in that pocket anymore, and I'll shut down that cancer cell. Now, I won't go through this because this is about 10 years of work here on this one slide. So this is what we did. And actually, I did it at a place called the Albany Medical Research Institute, right over there in Albany, New York, which is not that far from you. So what they had were these computers. And what I did was what we call a virtual screen. I actually used artificial intelligence to help me find a molecule that would fill that PCNA pocket. And this is how it works. they had databases composed of 6.5 million molecules, and now of the 3D structures. So they have databases, 6.5 million 3D structures of all kinds of little molecules. They come from mushrooms and plants and everything, okay? 6.5 million little molecules with their own 3D structures. I knew what the structure of that pocket was, right? You saw it as that pocket. And what we did was gave them the structure of that pocket. And for a week, think about how much computing time this had to be for artificial intelligence. 6.5 million compounds with the 3D pocket of pCNA went like this. And they were looking for compounds that could fit that pCNA pocket. And they came back to me and they said, Dr. Malkas, we found 8,400 of them, molecules. Now that is too many for me. 8,400 molecules for me to evaluate. It was too much. So what I did was, I won't go into the pain of this because it was pain, but I actually did a whole lot of study of that pocket and got really refined on what it looked like for its binding partners, all those other proteins coming in, what it really found. And with that information, we went back in, changed the artificial intelligence, redid the binding now of 6.5 million compounds into this really refined pCNA pocket. And we came up with what we call 53 candidate molecules. Very unique scaffolds. So I took them back and to the lab, 53 molecules, and I tested them between malignant and non-malignant cells. And of the 53 molecules, I found 5 that killed the cancer cells and left normal cells alone. Eureka. I found something. Now these molecules are very far. Yes, this is great. I've got 5 molecules that can kill a cancer cell and leave a normal cell alone. But they are hardly a drug. They're just they're like a skeleton of maybe a drug. I worked with my colleagues at City of Hope. We did a ton of chemistry trying to turn them into a drug. Eventually, we got them down to what we call some analogs, and then we got a candidate drug. So the candidate drug that we had was called AOH 1996. And this is what it showed. Very exciting. I can't tell you how much work to get to this point, but extremely exciting. So what you're looking at is, I'm showing you what they call cytotoxicity studies. And I'm looking at either non-malignant cells, which is in red here, versus malignant cells, and their ability to kill these 2 different cell types, and the ability of the drug to kill these 2 cell types. So what you can see is, so you're looking at increasing amount of drug, and this is the amount of viable cells. So you can see that AOH 1996 barely has any cytotoxic activity on those normal cells versus the cancer cells, readily kills the cancer cells. So there's a real selectivity now for the killing of cancer cells versus normal cells. This is kind of like what I said from the beginning, right? And then this is another cancer type. This is actually looking at lung cancers here. And the normal cells are red and blue versus the normal cells are red and blue versus the cancer cells, the lung cancer cells, which are in black. And again, you can see there's a real preferential or a real ability of AOH 1996 to selectively kill the cancer cells. We sent AOH 1996 to the National Cancer Institute in Washington, and they have what they call a 60 cell line panel. which represents all the major cancer types. And they themselves tested AOH 1996 versus all 60 of these cell lines. And they found that AOH 1996 had activity against every one of these cancers. So remember, I told you that that funny form of PCNA, the cancer-associated PCNA, we had found was a cancer-associated activity. Okay, that was something, a funny thing about cancer cells. And that the drug we have, or the compound we had developed, AOH 1996, selectively killed all these different cancer types. Now, one of the things you have to do, that I show the selectivity for killing cancer cells was really, really important, yeah, but does it really hit the target? Now, I worked with a wonderful investigator here at City of Hope, two investigators, Jeff Perry and his student, his young trainee, Jennifer Gossett, and they are what they call crystallographers, and these are brilliant, brilliant people. And long story, it took him 2 years to do this, and he would, so what they can do is they make these crystallized PCNA molecules, and they try to see where our little AOH 1996, where does it bind on PCNA? Because right now, everything's a model. But I needed the hard science to show that, in fact, it really did hit that pocket. And Jeff worked for 2 years on it. And my hat's off to him and to Jennifer. And what they showed was indeed the molecule of AOH 1996 really sits right in the pocket of PCNA. So AOH 1996 indeed was the snowball that fills up that pocket. And this is just, don't worry too much about this, but again, I want to show how cancer selective this drug is. These are called cytofluorographs. Don't worry too much about what they are, but they're just measuring cells in the cell cycle. These are non-malignant cells. These two bottom here are malignant cells. And you're looking at, time that we incubate these cells with AOH 1996. And you're just looking at the profiles. So here you have non-malignant cells incubated for 48 hours, and you're just looking for a change in their profiles of the cells. And you see essentially there is no change. This drug is not bothering normal cells at all. But the cancer cells, hot damn, look at them. They really screw up the profiles. And these part of the profiles here, these little white areas, are actually the growth of what they call apoptotic cells. It's killing, these are dead cells. Clearly, AOH 1996 is selectively killing cancer cells. This is now working our way towards the FDA. In order to do a clinical trial, we did a whole bunch of different tumor models. Here you're looking at breast cancer cells, breast tumors growing in mice, neuroblastoma, or pancreatic cancer. And what you can see is that indeed AOH 1996 is inhibiting tumor cell growth in animals. All right, pancreatic. So all these different cancer types. And we looked at the tissue, and all this is saying is that, in fact, we were showing that our drug indeed was getting into the tissue, the tumor tissue itself, and killing the tumors. Important thing as I'm moving towards, and I'm really glossing over. Remember, this is years and years of work. One of the important things is for it to be stable. And when you take a drug, you put it into syrup and it'll pass through your liver. And this is where a lot of drugs die, or would-be drugs die, because they have no stability in liver or in serum. And you can see that our drug has great stability in liver, as well as serum. The drug that we developed is an oral drug. I wanted to make an oral I wanted it to be easier on patients, that, they didn't have to be hooked up to infusion, that they could just go home with pill bottles. And because of the stability, the patient can take it twice a day. And that's what they do. We also performed studies, toxicology studies, looking at the organs of animals that have been treated for extended period of time with AOH 1996, and it was observed that there was no observed adverse effect on any organ, including the heart. So, summarizing, all right. I've talked to you about how replication, DNA reactive replication, is corrupted in cancer cells, that corruption is correlated with the advent of a funny form of PCNA, which we call the cancer-associated form of PCNA. I was able to build a snowstorm that packed up a pocket in that funny form of PCNA and acting like an airline terminal under a snowstorm, blocked the jets, the other proteins, from binding it. AOH 1996 has now entered phase 1 clinical trial here at City of Hope. It will be opened at other centers. I also have a trial that is in pet dogs, and you've probably been hearing my pet dog walking around here with her nails. But I have a trial that was opened at Case Western Reserve, and 57% of the dogs that pet dogs that were on that trial went into stable disease. So we also showed that you can target molecular hubs for anti-cancer therapy, and that there is no such thing as an undruggable molecular target. These are all the people that have been involved over the years in this work. These are the different funding mechanisms. And the little young one here, who I never met, I always say that when you see me, there is a 9-year-old girl sitting on my shoulder, and her name was Anna Olivia Healy, and her birth year was 1996. And remember, I wanted to do something bold in the name of that little girl, and that's why the drug is called AOH 1996. And as I said, I'm Alice. I followed the rabbit down the hole. I caught the rabbit, and our mission continues. So I want to thank you for your kind attention. It was so much fun getting to talk to you.
Thank you so much, Dr. Malkas. So
Thank you so much, Dr. Malkas.
So we do have a few attendees here today. If you are interested in asking Dr. Malkas a question, you can write it right into the question section on your GoToWebinar menu. Just click on that arrow and a screen will open up and you'll be able to type in there. Thank you so much to Bridget, who acknowledges with a brava for your presentation and your good work, Dr. Malkas. So, you've kind of brought us on your journey with you around the discovery and development of the new drug, AOH 1996. Tell us a little bit about, the timing on this, because if I'm a person who has had a diagnosis and I'm hearing this webinar, I'm going to be like, of course I want this. I want whatever is going to reverse my cancer and extend my life. What are some of the issues that were along the way? You told us about one meeting with this pharmaceutical company that said this is outside the realm of normal clinical chemistry trials and development. What else happened along this journey that felt like it was slowing you down or ended up having to be a diversion. You mentioned, being in FDA and looking at clinical trials. So tell us a little bit more about what it takes to really develop a new cancer drug that is so promising.
So there's, we could have a whole nother one just on this, on that. There's so, there's the building of the molecule, the stability. So I thought we had the molecule actually like 6 years ago. I had another, it's like the parent of AOH 1996. I thought that was going to be the one to go to the clinic. And I did all this stuff to show, oh my God, it's inhibiting replication in cancer cells, not in normal cells, repair. I did all of this work. I learned something very valuable that I know to do first now, as opposed to last the way I did it then. So this other molecule, which I called AOH 1160 at that time, I did all the science, everything nailed down. It was only then that we actually incubated it with sera with liver microsomes, and I discovered the drug got chewed up in 30 seconds. I'm not joking. That was like, had to go all the way back to the drawing board, figure out where the molecule was getting, where 1160 was getting chewed up, and then go and redesign it, so to speak, so that those sites that were getting chewed up by the sera or liver, they were not now, would not now be allowed to get chewed up by inside the humans. So that led, now I went from a third to 1996. So I went from a 30-second stability inside of a human to now for almost 5 hours. That's why the drug is given twice a day. Other things with the trial. So now it's in phase 1 trial, and CFO never wants me to talk very much about it because they say, don't talk about it. But so far, all the patients that have been on the drug so far, they have not shown toxicity or very significant toxicity. And that's, remember, that was the premise of the drug. So I'm very happy about that. But it's a toxicity trial. The way the process, and you all probably understand that, phase 1 trial is just to see toxicity. What's the maximum tolerated dose a human can take? I don't think we're ever going to find the maximum tolerated dose. So we start at, in the phase 1, you start out low dose, increase. So we're about 30% through our trial so far, and it's extremely well tolerated. The cycle is for 30 days, and so far, so good. All right. The next trials that come after this are what you call your phase 2 trials. I'm also trying to speed it along. There's something called phase 1Bs, because we have some other work that we've done in the laboratory as to how this drug could be positioned therapeutically. Now, for the longest time, everybody talked about monotherapy, right? It means one therapy, like for the longest time they treated either with a platinum compound or one kind of therapeutic. Those days are gone, or pharmaceutical companies are not going to like that, okay? Because they love one size fits all drug. The thing that we're learning through precision medicine and by the sequencing, the DNA sequencing of people's tumors are almost as individual as their fingerprints. And what we're finding now is that the best way to treat a person is actually by the creation of what we call combination therapy or cocktails, right? Everybody's tumor is different. So you're going to, by the genetics of the tumor, pick those therapies that are better for treating your tumor, not tumors, your tumor. Now, the thing that I'm finding with AOH 1996 in the lab is that our drug synergizes, the word synergize, it enhances the activity of a lot of the currently used therapies. It also allows us to lower the concentration of the currently used therapies. So we would make even them more tolerable in treating a patient. So I've been joking, you'll love this, Barbara. I call it, I'm eventually going to be the olive in everybody's cocktail. You know, AOH 1996 will be the olive in everybody's cocktail. So that's, you know, so AOH 1996, I don't think it will be used as a monotherapy, though my lab is trying very hard to make a cancer cell that's resistant to AOH 1996, and they have not been able to do that. And I think it's because we're not just shutting down one protein, we're shutting down technically 200 proteins. So it's very hard for a cancer cell to figure its way around. I don't know if this is helping you, Barbara. It's kind of like a long thing.
It speaks to the complexity, right? You started off talking about the complexity of our human body, our mechanism, our DNA footprint, and how in one limited section of that genetic map is an address that makes the world of difference to the mutation of cells within the human body. So it really does speak to the complexity of the science, but also the complexity of the solutions and therapies with respect to treatment and treatment options. So that is very helpful, I think, in understanding at least that much, right? That when a woman is facing a breast cancer diagnosis, she is in fact asked to make a lot of decisions in a very short amount of time regarding her overall care. And so that speaks to why those decisions are complex and, you know, multifold. You know, surgery, what kind of surgery, what kind of postoperative treatment, what will be follow-up therapies. You know, all of that complexity requires that a woman facing a breast cancer diagnosis needs to have a good care team and be well informed. So thank you for this work that you do. I do have one more question that I wanted to ask you, which was, you know, where are things at right now in this present moment? And barring any other timeline issues, how long will it take to get through this FDA process so that this is a viable therapy for folks to consider?
Yeah, no, it's a great question. So the traditional path is, you know, you go through phase 1, which is a toxicity trial. Then you go into what they call phase 2, which is an efficacy trial. And depending on how that works, they then do these huge phase 3. And I mean, it's a long time and a tremendous amount of money. But just for you and the audience here, what I'm trying to do, and I'm doing a tremendous amount of lab work right now, is I'm trying to thread a needle. The thing is to try to get your first indication. And so I'm trying to define, I know you're breast cancer, but I'm trying to define a cancer that would allow me to get to the FDA to get a positive indication as quickly as possible, potentially just after a phase 2 clinical trial. So actually, City of Hope has asked me this. They say, how long do you think it would take? Don't quote me on this, but I'm going to try my best to try and get through in the next 3 to 4 years. I know it seems like a very long time, but a lot of these, if I was to go through these big phase 3 things, you're looking at more than, you'd be like 14 years, okay? And I'm working very hard to try and go as quickly as possible. I'm hoping either we get an indication for monotherapy for a particular cancer, or potentially in a combination. Potentially the combination could go even faster. I've been to, I already see that our drug synergizes well with platinum compounds, with some of the topoisomerase inhibitors. So potentially it could be used as in a combination indication. So, but once you get your first indication, it gives oncologists leeway for its use. The other thing, a lot of people they go, I want to be on your trial. I want to be. The FDA will allow you to do that. Okay, you have to technically have been through all of standard of care. So all of the things that because it would be unethical, okay, to go onto a trial on an experimental drug if there are in fact other things that they know that can help you. So you technically have to have exhausted all standard of care before you could go on to a phase 1 trial. And that's for not just for our drug, this is for all drugs. So yeah, I want to give people hope. I think people are, I know somebody who has never gone into remission with breast cancer. She's 12 years out. All right, but they keep looking at her tumor, what changes, and they treat it. So good times are coming. They really are. We're much smarter than we used to be. Even when I was training, it was like, well, everybody was put on the protocol. Everybody had a single protocol that they went on, and that's not how things are now. And people actually, the whole cancer treatment area is being viewed very differently now. They're not looking at it as like, it's a breast cancer, it's a ovarian cancer. I mean, yeah, tissue type is important, but the way they're starting to really look at a cancer now is a molecular disease. So they look at the molecular makeup of your tumor, and that's what's driving the therapeutic choices and how your maintenance of it and everything else. So you're actually looking at a paradigm shift for how we've treated cancer for the last 50 years. So you're in the middle of a paradigm shift, very powerful one.
I have one last question, and then we will be quickly running out of time. And my last question is, answer as briefly as you can. So MBCC is an organization founded on the prevention of breast cancer due to environmental causes. This machine, the change that happens to pCNA that makes it more acidic, that impacts this machine that is in fact assisting in DNA replication. Is there research being done on a molecular level as to what factors are causing that change that may in fact be due to environmental exposure outside of our other concerns with respect to genetic disease?
So you can think of this as, I have really put a lot of thought into and a lot of work into this, is actually a stress response. Some other work that we have in the lab, and I can't have to be fuzzy on this because it is very new, but it is really very cancer cells are stress responses. They really are, if you look at it. Chronic inflammation has if you have a chronic inflammation of some sort, there's a higher risk for the development of cancer. This is a lot of data coming from all kinds of places. Every day you make cancer cells. 8 cancer cells every day. Every human makes them. For instance, before they were born. So your immune system is constantly taking them out. So things that strengthen your immune system. Okay, so it constantly recognizes malignant from non-malignant, takes out the malignant. Stress, and I think, good food, sleep, how you think. Believe it or not, they're doing a lot of study to show that stress has an enormous impact on our molecular makeup. I'm not going to say it's the only thing. I mean, you have environmental things, but you think of environmental things, these are all stress things.
Right.
And so the story is so far from, we are so complicated as organisms. But I think we can help ourselves a lot more than we think we can. And even when you have cancer, my observation is that the kinder you can be to yourself makes a big impact. So be kind. It's about stress, and like the funny form of PCNA is a critical. It's not like stress, like doing weightlifting, muscle stress. That funny form of PCNA looks to be a response to a critical stress response. Cancer is a critical stress response. Okay, it's the body turning on itself. It's a reaction, So,
and it could be a reaction to something in our environment as well as our own internal biome that is impacted by maybe the psychological and emotional stress, but also the physical stress of an environmental factor. So
and that's been long known. I mean, that's the environment. That's how. I mean, there's so much around that is known about that. Those kinds of studies are harder because you don't get it's not like like in the lab, you can get 100% of your assay to work when you've got people living eating all kinds of things, breathing all kinds of things, being subjected to all kinds of things. Very hard to do a definitive study. My colleagues who do this kind of thing all the time, they almost think I have it easier, and I think I have a hell life. But it's a very hard nut to crack to do these very large population type of studies and get a very definitive response. But there are things that we do know. I mean, there are foods, there are things okay, it's not 1,000% definitive. Yeah, there's certain things, additives and everything else. I mean, they have taken some things out of foods because they have shown that they're carcinogenic. So reading labels is not a bad thing. And knowing what to look for, and real food is good. Real sleep. In Europe they augment therapy. They augment it with diet. They actually take sugar out. I mean, in Europe, they're doing some very, very interesting nutritional studies where they really remove the refined they try to get the refined carbohydrate down to nil. And that's because of the metabolism of cancer cells. Cancer cells thrive on sugar because they have to divide all the time. Cancer cells are dividing, and they switch over. A normal cell uses protein to make its energy. A cancer cell uses sugar, okay, because it has to grow all the time. So sometimes, when I see, and parents meet some, children with their cancer, and they're trying to get them to eat, so they give them milkshakes, and I understand why they want to get something into them calorically, but the other part of me is like, so yeah, so there are, I suggest to people, I always suggest this, diet is really important. Diets, rest, I understand, Lord knows, that there are a lot of stress things that go on, especially now in people's lives. But realize, be kinder to yourself, be kinder to everybody around you.
That's a great note for us to wrap up on, about being kinder to ourselves and to those around us. On behalf of myself and the rest of the Massachusetts Breast Cancer Coalition Board, and Cheryl Osimo, MBCC's Executive Director, I want to thank you, Dr. Linda Malkas, for this very informative discussion today. And I want to thank all of our many listeners for joining us. For those interested, the recording of this webinar will be made available later today on the MBCC website at mbcc.org. If you would like to further support our mission of preventing environmental causes of breast cancer, please consider making a donation by visiting mbcc.org/donate. I appreciate your consideration. Have a good afternoon. Thank you again, Dr. Malkas.
Thank you, Dr. Malkas.